Genetically modified foods, also known as GM foods, are a common food type in the world today. But with its increasing popularity, there is also an emerging controversy regarding the harmful effects of it. In fact, there has been a huge amount of skepticism and fear among GM foods due to the public's lack of knowledge in the genetic engineering of it. One of the greatest connotations about GM foods is that they are mutated frankenfood and are extremely harmful to our health. But these myths have emerged due to the miscommunication and misconceptions brought by the public and media. To directly manipulate the foods we consume, many have voiced their concerns regarding the safety of GM foods, and rightfully so. Many have speculated that genetic engineering in GM foods are novel technologies, and thus is still unstable and unsafe to implement into our diets. Welcome to Demystifying Genetics. In today's episode, we will be uncovering the truth behind genetically modified foods in Canada. To better understand the true intentions and impacts of GM foods, we will demystify some of the specific myths that build up the miscommunication as to why people think GM foods are so harmful to our health. To really understand whether GM foods are as bad as everyone makes it out to be, we need to first start at the beginning and understand what GM foods really are. So what are GM foods? To understand the complex genetics behind this, let's look at these animations. Genetically modified organisms, also known as GMOs, are organisms that have had their genetic material, as in their DNA, altered in a way that does not occur naturally, such as by mating or natural recombination. Natural recombination is a natural process in which our genetic material is exchanged between two different organisms, leading to the production of offspring with a combination of traits that differ from those found in either of its parents. As such, genetically modified foods, or GM foods, are foods that have had their DNA manipulated and altered through a unique biotechnology called genetic engineering. Basically, how genetic engineering, also known as genetic modification, works is that it involves the process of using recombinant DNA, or rDNA technology, to directly manipulate and alter the genes of an organism. Since the production of GM foods uses novel biotechnology to manipulate the food we consume, many have voiced their concerns regarding the safety of GM foods, especially in how unstable and unsafe it may be to implement into our diets. And this is the basis as to why numerous myths about GM foods have erupted. However, many of these myths are ungrounded and false, so let's take a closer look at them. One of the major myths about GM foods today is that genetic modification and engineering is a new technology and should therefore not be used in foods we consume. But to be very straight, this is false, because humans have been doing genetic engineering for decades now. Ever since approximately 8000 BCE, humans have been modifying plants and animals. Back in the day, crops were constantly eaten by pests or damaged by bad weather. And as time passed on, the human population expanded, and the normal agricultural practice did not meet the demand anymore. To make bigger, higher yields of food, humans started to use traditional methods of genetic manipulation, such as selective breeding and artificial selection. However, humans at that time did not have any knowledge of genetics. Dogs were bred purely based on physical traits, hoping that these traits will successfully be passed on to the newborn puppies. Although this method was proven successful with many organisms over history, the chance of success was quite low, and these traits did not pass down to the offspring as often. It would take hundreds and thousands of experiments before a desirable trait was reached. This situation changed drastically in 1866, when an Austrian monk discovered the fundamentals of genetics. Gregor Mendel bred two different types of peas and identified the basic rules of genetic inheritance, which led to the start of genetic applications in food-modifying practices. In 1922, the first hybrid corn was produced and sold commercially. This was followed by the utilization of radiation and chemicals by plant breeders in the 1940s to randomly change crops' DNA in hopes of producing better crops by chance. After several decades, in the year of 1954, thanks to Watson and Crick, both of whom discovered the double helix DNA, our understanding of genetics improved significantly. From then on, genetic technology only got better, faster, and more accurate. Fast forward into the 1970s, two science researchers, Herbert Boyer and Stanley Cohen, successfully created the world's first genetically engineered organism. It was through this that scientists discovered the potential of using genetic engineering as a more precise and controllable alternative to selective breeding. Thus, in 1994, the first GM produce created through genetic engineering, a GM tomato, was introduced to the world. They became available for sale after studies evaluated by federal agencies proved them to be as safe as traditionally bred tomatoes. These tomatoes were modified to include a DNA sequence that inhibited production of a natural tomato protein, increasing the firmness and extending its shelf life. So that is one myth busted. But before we move on, let's do a small quiz to test your knowledge. Fact check number 1. For your first question, after everything you have learned so far about GM food, which statement is true regarding GM foods? The correct answer is D. GM foods are food that have had their DNA changed by biotechnology. We hope you got that one correct. Make sure you are keeping track of your score for future fact check questions. Let's now move on to the next myth. Because many of us are unfamiliar with the term genetic engineering, people have a common skeptical impression on the safety of GM foods. This invokes another myth, where it is commonly thought that GM foods are dangerous because they can change your DNA. However, this can't be further from the truth. To understand why, let's first explore what DNA is and how DNA in GM foods are managed by genetic engineering through this animation. DNA are small molecules named A, T, G, and C that contain the basic hereditary material in our body. They make up a gene which carry all the information an organism needs to carry out vital functions. Genes are responsible for producing proteins that make up our muscles, organs, and skin. Through mating, these genetic variations can be inherited and expressed in a variety of different combinations by the next generation. Let's take a look at Mendel's pot experiment to understand the basis of genetics. Here is a yellow pod seed, and here is a green pod seed. The color of a seed is determined by a gene which has two alternatives. Let's call them big Y and little y. These gene alternatives are called alleles, and the combination of a pair of alleles within a seed determines its color. If the pea has at least one big Y, it will show a yellow color. But if the pea only has a little y allele, it will be green in color. In this case, because the presence of the big Y masks the little y, the big Y is called the dominant allele, and the little y is called the recessive allele. When an organism has one per allele and expresses the dominant type, this organism is called a heterozygote. So what happens if two heterozygous yellow peas crossbreed? This is best explained by a Punnett square, a square diagram invented by a British geneticist named Reginald C. Punnett in 1905. During crossbreeding, each parent passes one allele per gene to an offspring. Therefore, the combination of alleles an offspring may get can be calculated by a Punnett square as shown. From here, we can see that when the two heterozygous yellow peas crossbreed, there is a 3 out of 4 chance that the baby pea has a yellow color, while there is only a 1 out of 4 chance that the baby pea has a green color. Before scientists invented genetic engineering, there was no reliable method for selective breeding a certain trait, and people had no way of knowing whether the trait was dominant or recessive. As shown by Punnett squares, if the desirable trait is recessive, it has a rare chance of being selected and expressed, which poses greater barriers to selective breeding. Therefore, the invention of genetic engineering provides a massive confidence in a more accurate, efficient method of producing desirable food products. Before we move on to the different types of genetic engineering methods, let's do another fact check. For this question, what did Gregor Mendel do, and why was he so important? The answer is C. Mendel, through his pea-pod experiments, discovered the basis of genetics, allowing genetic technology to get to the level of genetic modification it is today. Now let's get into the actual process of genetic engineering and how scientists make GM food. Genetic engineering can select a specific trait and introduce it into the plant directly. To make GM food, scientists have to first identify a desired trait of interest, then to find an organism with that desired trait in its genes and isolate that trait. Next is to copy and insert the gene into the DNA of the new organism. And lastly, grow and breed the organism with the new trait. It's important to ensure that the propagated organism is monitored and tested in small then large field tests. In fact, GM foods go through in-depth review and safety tests before they are sold to farmers. However, it's important to emphasize that regardless of this advanced and novel technology, during genetic engineering, the new trait does not change existing traits in the plant or animal. Specifically, throughout decades, scientists have invented ways to genetically engineer DNA in living organisms. Here, we will introduce the three most popular methods they use in foods. The earliest technique was invented using a soil bacterium called Agrobacterium tumefaciens to infect the plant with the desired gene, which makes the resulting plant express the trait. In nature, this bacterium naturally inserts part of its DNA into a plant cell to make the plant a new sweet home for itself. Scientists discovered this bacterium and used its natural behavior by replacing the bacterium gene with the gene we want to see in the plant. This method is highly successful in certain plants, such as potatoes and tomatoes, but can be inefficient when used on other plants. The second and most widely used genetic engineering method is directly transferring DNA into a plant through microparticle bombardment, also known as the gene gun. This method was developed in the late 1980s by John Sanford. In the first step, tungsten or gold particles are coated with the gene we want the plant to have. Millions of these particles form a gene bullet, which is placed in front of a gene gun. When the gun is fired, the particles are propelled through a filter plate and then puncture the plant tissue, usually embryonic tissues from the seeds. As the particles reach the cells, the genes coded on the particles dissociate from the particles and get expressed in the cell. When the plant grows, the inserted gene is then stably expressed as a trait. This is a great method when we want to transfer larger genes or a large DNA fragment of multiple genes. There have been many successful examples using this gene gun technique. Some examples include soybean, wheat, and onion. The latest genetic engineering technology available is the CRISPR-Cas9 system. This technology directly edits the DNA of the plant. Similar to the method using soil bacterium, CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. Like a working set of scissors and glue, when a CRISPR-Cas9 complex is injected into a plant cell, this complex identifies a targeted site on a DNA string and cuts it open. The desired gene is carried by the complex and then inserted into the genome at the cutting site. The inserted gene is connected to the plant genome by the natural glues that exist in the plant cells. As a result, the gene is expressed, and the trait will be shown by the plant as it grows. This method is very specific because, unlike the other two methods, this can choose where it wants to insert the gene and make sure it's expressed while it does not affect the function of other genes. This method has already been used in many plant species, including cucumbers, apples, and papayas. In all three methods, DNA is edited with natural genes that already existed in nature. Two of the insertion methods are adopted from natural molecular mechanisms as well. Studies over decades have been investigating the safety of GMO food, but no clear evidence of them indicated a danger in human health or a risk of DNA modification through consumption. The name of genetically modified food may sound like the DNAs in the food are unnatural. However, every food you eat that contains protein contains DNA, whether that is GMO or not, thereby not affecting your own DNA. This is because enzymes break apart the DNA in your gut and it's no longer functional. In fact, when a crop is processed down to its sugar or oil, there are no proteins left. No protein means no DNA. In conclusion, being genetically engineered does not essentially mean the food is unnatural, and it's impossible for GM foods to change your DNA. That's another myth being busted. That was probably a lot to take in. So let's do another fact check question to test what you have been learning so far. Which of the following choices is not a genetic engineering method for genetically modified foods? The answer is D. Inserting a gene trait by injecting viruses is not a common method used for GM foods. We hope you're enjoying these fact check questions. Stay tuned for one more. With the increasing popularity of GM foods, another common myth is that GM foods are everywhere. In reality, there are only a limited number of GM crops being approved in Canada. Besides, being approved for sale does not mean a GM crop is allowed to be commercially grown in Canada. Health Canada has been assessing GM foods for more than 20 years with strict regulations. In fact, only 4 genetically modified crops are widely grown in Canada, and a 5th was introduced in small amounts in 2016. As of 2020, Canada also produces GM salmon. Any other GM crops are imported to Canada. These GM crops are mostly genetically engineered to be insect resistant or herbicide tolerant and end up as processed food ingredients in animal feed. In fact, many commercially available food products contain ingredients from GM crops. In Canada, it is not mandatory for GM foods to be labeled for sale. However, some food companies choose to label their products GMO-free. Labeling of GM foods is challenging. This is because GM foods like sugar, canola, and corn are widespread in our food supply. GM corn can be used to make high fructose corn syrup, which is used in other foods like cereal and pop. As a result, all of these foods would need to be labeled as having GM ingredients. One type of GM food takes decades to develop, be tested, reviewed, and approved. In the process, lots of studies and experiments have to be done to ensure its safety and practicality before being commercialized. Therefore, the main idea is that not everything we see is abnormally modified, since there are clear health and environmental regulations. And that is another myth busted. Before we visit our final myth, let's do one last fact check question. Which of the following GM foods is not approved in Canada? The answer is A. Bananas are not a GM crop that we produce or import to Canada. So how many questions did you get correct? Are you a demystifying genetics geek? We hope these fact check questions kept you intrigued. But let's move on. Another common myth is that people think nutrient levels in GM foods are significantly altered or lowered. However, this is not the case. In fact, numerous studies have shown no nutritional differences between commercially available GMO and non-GMO foods. For example, in 2016, the National Academies of Science, Engineering and Medicine, or the NAS, issued a report confirming the safety of GMOs as well as their compositional and nutritional equivalency with non-GMO foods. In another meta-analysis of 698 reports, it was found that all of the research concluded that in terms of health, there have been no observable differences between GM foods and conventional foods. Thus, it's important to note that even though in GM foods we are selecting for a certain modification type, this does not mean that it automatically changes the entire composition of the crop. Yes, in some cases, unintended consequences might occur, but this is why we have additional safety and nutrient testing regulations all over the world. Genetic modification can also improve the nutritional content of some foods. For example, golden rice is a GMO rice crop enhanced with vitamin A to increase circulating vitamin A levels in the blood to help combat irreversible childhood blindness, where vitamin A deficiency is the main cause. Despite the controversy surrounding it over the years, the benefits of this modified crop is plenty, where it has the promise to help prevent millions of deaths and alleviate the suffering of children and adults with vitamin A deficiency and malnutrition in developing countries. In fact, after numerous health and environmental testing, the FDA and Health Canada has approved golden rice for cultivation and declared it safe for consumption. This is just the beginning in the advantages provided with GM foods, thereby busting this myth as well. This brings us to the end of our episode on demystifying genetically modified foods, where we tackled some of the most frequently raised questions and concerns regarding the topic. As we have explained, many of these concerns are merely myths that have arisen due to the lack of knowledge about genetics and science. We hope that through this episode we have dispersed some of the burning questions you may have had towards GM foods. We will be catching you later in another episode of Demystifying Genetics, so stay tuned.